shout out, and Beth can also alert me to lingering questions. All right, so our first planet Microbes, a whole whopping 6 months ago, was also about viruses. And I was thinking, probably the entire planet has been thinking nonstop about viruses for the past 6 months, but only obviously one particular virus, and one that has really, it goes without saying, devastated everything, I guess. But viruses infect everything on Earth. Everything living has a virus that infects it, whether it's been characterized by science or not. And one of the interesting things about environmental microbiology is that it's not all about it's not all about death. It is about death, but it's also a complicated interaction that changes the way that the geochemical cycles of Earth work. And so I would say that for this planet Microbes, we should challenge ourselves to think about viruses a little bit differently than we normally do, and think about how they interact with the environment and are agents of environmental change and restructuring. So it's a little bit of a different lens shift, but I think you'll see that in the environment, viruses aren't always a negative thing that brings death and despair, unless of course you happen to be the cell or the critter that's infected. So viruses infect everything, and that includes everything on planet Earth. And of course, we think most often of people getting infected by viruses. This is Tom Hanks, a COVID-19 survivor. And so, we think a lot about what viruses do when they infect us. They make us sick. They shut down planet Earth. But viruses also infect a huge, they infect everything. So sequoia trees are infected by viruses. Fire ants are infected by viruses. And we can start to think about what different effects viral infection has on something like a sequoia tree or a fire ant relative to something like Tom Hanks. As we take a tour around the planet, they infect moss piglets or tardigrades, which are in the news all the time. They even infect the tiniest plant cells on Earth. This is a micrograph of Prochlorococcus, which is the most abundant photosynthetic cell on planet Earth. And this brings us back to the moving planet Earth on the right. And you can see in this false colored image from NASA, the red colors, all of the colors are a proxy for chlorophyll, or the stuff that makes plants green. And you can see that as the seasons go by on Earth, the planet is sort of breathing, and the abundance of different photosynthetic cells is increasing and decreasing. And in the ocean, the majority of these cells are these tiny little microscopic cells of Prochlorococcus. And so you can start to think, if a virus infects Prochlorococcus in the ocean, and these organisms are so important for producing the oxygen that we breathe, what effects do viral infection in Prochlorococcus have on planet Earth relative to, a virus infecting Tom Hanks? I should also point out that viruses even have viruses themselves. So there's this one particular type of virus called Sputnik that can infect other viruses. So it needs to hijack its way like a satellite onto viruses that are onto another virus that's infecting a cell, or else it doesn't work. So viral ecology and how these things interact with the host that they're infecting is way more complicated than just the way that we think of humans getting sick with the flu or COVID-19. And so we are looking today at viruses in the environment and thinking about why do environmental scientists care about viruses just as much as epidemiologists or people who are interested in human health? So to start out with an overview of how viruses do their thing, the viral life cycle when it comes to single-celled organisms. So viruses have two different strategies after they infect. So starting out here, a virus particle that looks like a little lunar lander will land on a cell, insert its genetic material into the cell. And the important thing to keep in mind is that these viruses are obligate intracellular parasites. And this means that viruses are not technically alive. They can't do anything without a host cell. They can't reproduce, they can't metabolize. Viruses can never move of their own volition. They require a host cell. Without a host, a virus is just a husk of protein with some genetic material inside. Exactly, like nesting dolls, except nesting dolls that can kill you. So viruses land on a cell, they infect it, they put in their genetic material, and from there, they hijacked host metabolism. And that can go either two ways. In one way, viruses integrate their genome into the host genome, and then they hang out in waiting. In a corollary with human viruses, this is like the herpes virus. You don't always, if you have the herpes virus, express that in the form of a cold sore or something, but some environmental trigger will trigger that virus to start producing. It will leave where it's integrated in your genome and then start producing more viruses. Or the virus can go another route and immediately take over host cell metabolism and start producing more viruses. That host cell then erupts and releases those viruses into the environment. And then the cycle continues when that virus progeny goes on to infect other cells. And then, of course, there are different flavors of viral infection that you can think about and how they might infect the environment, how long it takes this whole process to happen, whether viruses are released chronically or whether it kills the cell immediately is another thing to consider, and where the virus genome is integrated if it's sort of lying in wait. And all of these things will have different effects on how the virus is impacting host metabolism and also the environmental impact, which we'll talk more about later. So this is a good overview of what's going on with the virus, but now we have to consider what's going on with the host cell that the virus is infecting, and what are the environmental consequences of one strategy of viral infection versus another. So, like I said before, viruses are required to reprogram host cell machinery to take it over and reprogram them to make more viruses. And this is critical because viruses can't do it on their own. So this is a comparison of normal host cell metabolism, here looking at a photosynthetic cell, like I mentioned earlier, and thinking about what those cells do on a normal basis. They take in sunlight from the sun and use that to produce energy through photosynthesis. They use that to fix carbon that they use to produce more daughter cells. And along the way, they're taking in nutrients and fertilizer from the environment in the form of nitrogen and phosphorus. And their whole goal is to produce more daughter cells. So to divide and to repopulate. When you're infected by a virus, however, things get switched around. So what activities do you need to take control of if you are a virus infecting a host cell? And this is a diagram, a corollary diagram that shows how viral infection impacts host cell metabolism. So you might Now all of a sudden you have to turn this host cell into a little machine that produces more viruses. So you might want to uptick photosynthesis so that you can ensure that the host cell has enough energy to make more of your children viruses. You might want to increase amino acid synthesis, so making more proteins, which you will need to assemble more of those child viruses. You'll need more phosphorus to make more of the genetic material. But you might turn off some of the carbon fixation genes, because if you're going to kill this cell anyway, you don't care if they can fix carbon anymore. So they're reprogramming the cell to move away from making more of itself to making more viruses. And along the way, in the case of photosynthetic cells, that might change what the output of this cell is. So in normal photosynthetic cells, they're taking in carbon dioxide, a greenhouse gas, and producing oxygen. And how might that change if that cell is infected by a virus and things are getting reprogrammed?
Before you go on, Kyle, Tina's got a good question in the chat. She says, if host cells are already hospitable, does the newly infected cell change to become less hospitable to new viruses?
As like a way of like an immune response sort of that protects them? Yeah, single-celled organisms in the environment definitely have an immune response to viral infection. It's much simpler, obviously, than humans with T cells and because we have multiple cells. But one particular virus immune response is CRISPR. So we hear that in the news a lot, the CRISPR gene editing technology that can sort of serve like a Microsoft Word cut and paste tool that scientists can wield almost as easy as Microsoft Word to change genomes. So that is actually technology that was taken from bacterial cells' immune systems to viruses. And it works by taking a little snippet of virus DNA or RNA and then remembering that and killing the viruses if they try and reinfect. And so the CRISPR sort of is like a flip book of memory encoded within that bacteria's DNA. We probably should do a whole Planet Microbes on CRISPR in the environment, but actually the last author of this paper that we're reading was Jill Banfield, was heavily involved with basically telling Jennifer Doudna, who turned CRISPR into gene editing program. She basically told her, you should look into CRISPR. I think it could be used for this. So that's just an interesting point of science history. But there are other ways that single-celled organisms can evade viruses by having like a thick mucus layer that we've talked about a couple weeks ago or a couple months ago. So they can prevent viruses from actually getting to the cell and infecting. They can also just be very numerically abundant and hope that that viruses don't infect. And another strategy is that they can trigger what's called programmed cell death pathways. So one virus will infect one cell, and then that cell will go, oh crap, I'm infected. And it will release a chemical signal that warns all of its neighbor cells, its sisters, its daughters, that they've been infected. And then all of those cells will sort of altruistically kill themselves. And that will create a zone of death around this colony or these cells in the environment that will prevent the virus from spreading. Why does this work? Because, like I said, viruses are obligate intracellular parasites. If everyone they can infect is dead, there's nothing they can do anymore, and that infection will die out. So yeah, there's lots of different ways that host cells can make themselves less hospitable, but it's not always effective. It's a numbers game, I guess. Okie dokie. You move the chat and move the Brady Bunch. Title credits. Okay. So viruses are reprogramming host cell machinery to make more of themselves if they can get past those immune systems that we just talked about. So there are other ways that viruses can reprogram host cell machinery to serve their own nefarious goals, and that is by stealing genes from their hosts. And these are called auxiliary metabolic genes. And we read about these in the paper that we read for today's discussion. And these were discovered as sort of weird genes that you wouldn't expect to be in a virus genome. So one of the first discoveries of this were bacterial photosynthesis genes that lived in a virus that infects cyanobacteria. And this is weird because viruses certainly don't photosynthesize. So why would a virus that is infecting a host want to carry around these photosynthesis genes? We can talk about that a bit later. As soon as they discovered this, they looked around for this other places and found that photosystem genes, the genes responsible for photosynthesis, were actually pretty widespread in viral genomes, especially in the ocean. And this sort of opened up this field of these auxiliary metabolic genes. They looked in the deep ocean at deep sea viruses, and they found sulfur oxidation genes. And then they looked in hot spring viruses and found that archaea there include nitrogen metabolism genes. And does anyone know what all of these different classes of genes have in common? This may be a tough question, but what they have in common is that they're all used to generate energy for their hosts. So photosynthesis obviously is what plants use to make energy and grow, and other bacteria, other microbes use sulfur oxidation and nitrogen metabolism as sources of energy to grow. And the sort of working hypothesis of these auxiliary metabolic genes is that viruses are shutting everything down in the cell, but they still need energy. They can't do it themselves. So even though they've shut down host cell metabolism, they are encoding these particular genes that enable the host to keep doing what they do just for enough time that the virus can replicate itself and then hightail it out of there, kill the host, and infect more viruses. Does that make sense, or does anyone have any questions so far? Thumbs up, thumbs up. So far, so good, great. So I found this, when I was doing some background reading for this class, I found this review paper that had what I think is a really interesting and funny metaphor for how these viruses are working in their host cells. They can choose two different things to do. They can pillage and plunder or batten down the hatches. So this little sinking ship or floating ship is a host cell, cyanobacterium, photosynthetic cell in the ocean, and these little green or little yellow phage are the viruses. So there's two strategies. Either viruses can infect and they can say, you're gonna die anyway, we're just gonna make as much virus as we possibly can, as quick as we can, we don't care about you, and then they are finished and they kill the cell, and this poor little cyanobacteria sinks. On the other side, they can batten down the hatches, and they can use these auxiliary metabolic genes to keep the cell happy enough so that it can produce as many viruses as humanly possible, as cyanobacterially possible. I'm anthropomorphizing these because they have faces in this picture. So they they will encode a photosynthetic gene or some other energy generating gene and say, you know, things are fine for now. You're still going to have energy. And we will make as many new viruses as we possibly can. What this image doesn't show is that after they batten down the hatches, eventually the viruses get what they need and they will end up killing the cell. So ultimately, the ship sinks, unfortunately, in most cases. And then the last thing I wanted to talk about is that in addition to reprogramming the host cells, viruses also sort of reprogram the environment. So phytoplankton in aquatic systems, those are at the base of the food web, and they are just like trees and plants. They are producing the food that fuels every other organism up the food chain. So in this image, phytoplankton are feeding tiny microscopic grazers that are food for carnivores. And then you can imagine this going all the way up the food chain to humans or to sharks or whales or whatever your favorite charismatic megafauna is. But when viral infection happens, they are sort of shunting all of that energy into to smaller compounds that are no longer accessible to these larger organisms. So this so-called viral shunt is like sort of like chutes and ladders. It's moving all of this potential energy right back to bacteria, where it sort of stays in this smaller zone and can no longer support these larger organisms up the food chain. So this sort of comes up with the question of how viruses using these auxiliary metabolic genes might impact the environment, depending on what those particular genes are. And that brings us to the paper that we discussed today about how large phages or viruses that live in lakes have the potential to alter methane oxidation. So before we get started talking about this paper, does anyone have any questions or comments so far? Great. So we'll just go through this paper and then please stop me again if you have any questions, and then we will break out into discussion groups, and then we'll all come back together at the end and have a chat again. All right, so this paper was published 2 weeks ago or last week in Nature Microbiology, and this paper takes us to first a methane generating tailings lake in Canada. And so this is a picture of the site that they took their samples from in Canada. As you can see, this is a lake, it's a man-made lake that's right on the shores of a factory. And so these lakes tend to be very polluted and not very hospitable to most things, of course, with the exception of microbes that live everywhere. And these lakes, because of the conditions, the geochemical conditions within them, they have, they're home to organisms that produce lots of methane, or these organisms are called methanogens. And methane that's produced in the sediments of lakes, it can bubble up into the atmosphere, and this is bad because methane is a greenhouse gas. It's not as abundant in the atmosphere as CO2, but as a greenhouse gas, it's 20 times more powerful than CO2. So its ability to trap heat on Earth is much stronger than CO2. So we have a vested interest in keeping methane emission from lakes like this down. Of course, for every microbe that produces something, there's another microbe that eats that thing that they produce. So in these lakes, methanogens are producing methane, but there are also methanotrophs that consume or oxidize methane for carbon and energy. So just like humans eat carbon that's produced by plants for our energy, these methanotrophs are oxidizing or using methane, which has the formula CH4, as their carbon and energy source. And they do that using 3 enzymes that are responsible for methane oxidation. These are the PMOC, A, and B genes, and they are clustered together in the bacterial genome on an operon, or sort of like a sequential set of genes. And these genes make enzymes that turn methane into methanol, and along the way they are using carbon and producing energy through this process. So both of these organisms are existing together in this lake, and the methanogens are producing methane and the methanotrophs are consuming methane. And so you can imagine that the relative proportion of one versus the other will control how much methane is released from these lakes, which of course goes on to warm the planet more or less. So now, where do viruses start to fit in here? If viruses are killing one or the other, then they're changing that proportion of how much methane is potentially released. So what will happen if viruses encoded these methane munching genes, which is what this whole paper is about? So they went to this lake and they discovered that certain viruses that live in this lake actually encode these bacterial genes that are responsible for the breakdown of methane. And so they went about to figure how these might be used, if they're used, and what the potential consequences might be for methane emission from these lakes. So these researchers went to this lake in Canada, and they took samples over several years, which you can see going across the horizontal axis here. And they tallied the amount of methane that was produced as the years went by. So Beth has a great question about whether they knew for sure that they were from viruses. So they knew that these were from viruses by, we'll get into this in a bit, but they knew that they were from viruses by looking at the complete assembled genomes from which they came. So you would be able to tell that was definitely not a bacterium or some other microbe. But we'll talk about that in the next slide. Exactly, yeah. So they went out in several years from 2015, and they took samples to look at how much methane was produced in the lake, and that's in these black bars. So some years have more methane than others, and they also looked at the abundance of methanotrophs, so the bacteria that are eating that methane. And the different colors in these sort of reverse vertical bars are the different flavors of methanotrophic bacteria that they found. So mostly these purple gamma proteobacteria and these orange gamma proteobacteria. And they also found varying levels of these different methanotrophs as the years went by. And these stars indicate where methanotrophs were less active. So in the years where there were fewer methane-eating bacteria, they also saw more methane, which makes sense. So this gets back to Beth's question, what they did. If they wanted to explore the genetic potential of the viruses that live in this lake, they could really only do this using a genomic approach, because that is one of the easiest ways to study all of the viruses, because otherwise they would have to isolate the hosts, and then isolate the viruses and then grow them up a lot. Again, because like I said before, these viruses are intracellular obligate parasites. Without their hosts, they're nothing. So the best way to study what they are and what they can do is by looking at what is in their genomic toolboxes. And they do this using an approach called metagenomics. So you basically, to simplify this, you take a bucket of whatever your sample is, extract out all of the genomes, all of the DNA from within that sample, and then you cut that into tiny pieces, and you run that through a sequencing machine, all mixed together. And then you can use very powerful computers to put together all of those pieces like a jigsaw puzzle, and use more computers to put those into different bins. And then ultimately, you can, when you have a finished picture of the jigsaw puzzle, you can hold it up and look at what that picture makes. So in the case of it being viruses, there are hallmark viral genes that you can look at and know that it is a virus. And the same goes for bacteria or other microbes. So that's what these scientists did in this lake to look at the different viral communities. I'll stop here again and ask if anyone has any questions.
I have a brief one. When you're doing this metagenomics, do you get rid of all of the bacteria and only look at the genomes of the viruses before you do this analysis of the different kinds?
That's a great question. There are some techniques that you can do to only look at one fraction or the other. So viruses are really small. So much smaller than bacteria. So you can filter your sample through a 0.02 micrometer filter. It's basically the smallest coffee filter that exists. And the only thing that should be able to get through that is a virus. And then you know that you have viruses. You can also look for the genomes within the host cells. So that would show you all of the viruses that are actively infecting host cells at that time. So you would get the genetic material from the viruses that are actively making new copies of themselves within host cells. Or you could use certain computer algorithms that can pick out specific viral genomes from within the melange of everything. So it just depends on realistically how they took the samples when they were out there floating in the middle of the lake or wherever you're taking samples to figure out what would be best. But where there's a will, there's a way, and they'll be able to do it.
Thanks.
That was a great question. Any other questions or comments? Okay. So these researchers went to this lake where they knew methane was being produced. They sequenced these metagenomes, and then they figured out that there were some very large viral genomes in the lake that happened to contain this PMOC gene, which is one of the critical genes in methane oxidation. There's no part of viral metabolism that should require methane oxidation. So they assume that this is one of those auxiliary and metabolic genes that must have some critically important, must be something that is critically important to these, to infecting host cells to enable the viruses to do what they do. That's a good hypothesis, but they first had to figure out, is this really actually a PMOC gene, or is it just something that looks similar to PMOC? So to do that, they compared PMOC genes from bacteria that are well characterized. We know that these proteins are actually doing the work of converting methane, oxidizing it into methanol, and they compared that to the sequences that they recovered from these potential viruses. So in this figure here, we have what is called a protein alignment. So each of these horizontal bars is a different sequence. These three on the top are from PMOC protein sequences from bacteria. We know what they do, they're confirmed. And these four sequences on the bottom are the genes that are recovered from their viral genomes from this lake. And where you see green up here along the top are regions where they align completely. There's perfect correlation between the bacterial genes and the bacterial proteins and the viral proteins. What is important to note about this alignment and how these bacterial and viral genes compare is that well, first of all, they look really similar overall, but also in these key important places where the enzyme is actually doing the work, there is really strong alignment. So all of these sequences have the same amino acids at this position. And this is pretty strong evidence that this PMOC gene, it's not just some random gene. If you turn this into an enzyme, it would actually do the work of converting methane into methanol. They also looked at who these PMOC genes are likely coming from, what host. So they made what are essentially family trees out of these genes and compared the hierarchy of how these genes compare to various bacteria that have these genes. So this is a zoomed in version of these family trees with the viral genes in green and yellow and the bacterial genes in black here. And you can see that they are actually closely related to the bacteria that are present in this lake and genes that and they come most likely from bacteria that are the hosts of these viruses. So that's a good indication that the viruses have stolen or co-opted this gene from their hosts. What they did next was look around to see if this was just one particular special instance of this of viruses having this auxiliary metabolic gene in this particular lake, or if this was widespread in methane emitting lakes around the world. If it only occurred in this lake in Canada, then it probably wasn't super important for global emission of methane from these types of lakes. But if it was present in multiple different lakes, then this is a good indication that this is a common strategy used by these viruses, and that it could be something that needs to be considered as an important facet of the methane cycle on Earth. So this is a super complicated figure, but I'll walk you through it here. So they went to different lakes in Switzerland and in the United States and compared viral sequences from those methane emitting lakes to the virus that they had previously been characterizing. So each of these horizontal lines are all of the genes within the various viral genomes that they have found. And this green horizontal bit shows you all of the viral genomes that have the PMOC gene. So a handful of these related viruses have the PMOC gene, not all of them, some of them don't. And then you can see various other potential auxiliary metabolic genes that these viruses encode. And what is the takeaway from this image and this analysis is that there's not a lot that these viruses have in common. And for me, this indicates that if one thing that many of them have in common is this PMOC gene, that means that this is something that is potentially conserved in these viruses. It's something that they're keeping around. They're not getting rid of it through evolution and adaptation. And this is, I think, a good indication that this capability is just as important as the genes that enable them to replicate their DNA or to make more virus particles. And then finally, the final question is, okay, these viruses that live in these high methane lakes, they have these PMOC genes, they're widely distributed, but do they actually use them? If they're just carrying around these genes and they're not actually using them, then again, it doesn't really matter if they have them because it's not going to impact the carbon cycle or the amount of methane that's emitted. And so what these researchers did is they looked at whether or not these viral PMOC genes were being expressed. So you start out with DNA. Before you can get to proteins and these viruses actually using them, you have to go through RNA. So transcription and then translation. And so they instead of sequencing all of the DNA from these samples, they sequenced all of the RNA. And that enabled them to see if these viruses were actually turning on these genes and ultimately translating them into proteins. So they sequenced all of the RNA genes, or they sequenced all of the RNA from these samples using metatranscriptomics, sort of the same thing as metagenomics that they used earlier, but with RNA. And what they found is that indeed, these genes were being expressed by various different viruses through time. And this is a great indication that these genes are actually active and being used. And if they're active and being used, that means that they are doing something with methane and impacting potentially the amount of methane that is released from these lakes. I think the final interesting thing that they found is that the bacteria that were thought to be infected with PMOC containing viruses had the fastest growth rate. And translated into a different way to say that is that the viruses that were infected by I was trying to make it simple and then I messed up my host and virus. The cells, the host cells that were infected by viruses that have PMOC, those are the sinking ship. They're on their way out. But they had the fastest growth rate. And this could be an indication that these viruses are using PMOC, even though they're on a sinking ship, to extract out all of the energy from their host cells through methane oxidation that they possibly can while they're trying to make as many daughter virus cells as they can before ultimately they jump ship and go on to infect other cells. So that's the final tidbit of evidence that these viruses are using these genes to co-opt their host metabolism, and that this could have an impact on the methane cycle. Of course, this makes me ask the question of, that's amazing, but what's next? The one thing that they didn't really look into in any great detail in this paper is assessing how much is methane reduced in a population of cells that is being infected by these viruses. Could we potentially use these critters as a way to control methane emission in these really polluting lakes? So maybe that's a follow-up question that they have to go into the lab to answer. But that brings us to the discussion portion, and maybe we can talk about that more in our breakout groups. So before we beam each other to the breakout groups, does anyone have any questions or comments or things that weren't clear or food for thought? Usually these go in pretty shocking directions. So if there's anything, anything at all anyone wants to talk about. Oh, that's a great question, Stephanie. So Stephanie asks if the virus might be able to introduce methane oxidation abilities into a non-methanotrophic bacteria. This is something that I didn't touch on that I probably should have. So one thing that viruses can do, not only can they take genes from their hosts, but they can also transfer genes from one host into another. So viruses are also agents of evolution and giving new capabilities to potential hosts. So 100% definitely they could. The problem is that you really need the entire PMO, CAB, I might be forgetting the exact letters, but you would need all 3 enzymes in that cassette to do everything. But they could certainly give the host that PMOC gene. Whether or not the host could use it to oxidize methane and have suddenly a new energy yielding capability, maybe not. You would also be limited by the host specificity of the virus. So just like the novel coronavirus needs to use its spike cell or spike protein to infect our lung cells, that's a highly specific interaction. So the virus would need to find a new host that isn't a methanotroph, that has a similar point of entry. So I don't know how common that would be, but certainly viruses are spreading genetic. They're constantly taking up their host DNA, whether it's into their own genome or repackaging little bits of stuff that's floating around the host cell. We could have a whole nother class on that too. It's a super, super fascinating topic.